Hello my dear students, welcome back to Biology at Priyajit. Have you ever wondered how your kidneys can filter nearly 180 liters of fluid every single day yet produce only about 1 to 1.5 liters of urine? How does the kidney know what to keep inside the body and what to remove? The answer begins with one of the most important physiological processes in the human body, that is glomerular filtration rate or GFR. In today's class, we will understand what is glomerular filtration, what is GFR, glomerular filtration rate, why it is called as ultrafiltration, what factors regulate GFR, and why this GFR is considered the best indicator of kidney function. Let's begin. Introduction to urine formation. The kidneys receive approximately 1300 ml of blood every minute, which is nearly 25 to 60% of the cardiac output. The primary function is to remove waste products while conserving useful substances. Urine formation occurs through three major steps. Number one is glomerular filtration, that is glomerular capsule, and this is the filtration from blood plasma into nephron. Blood plasma is filtered into the Bowman in Bowman's capsule of the nephron. This is the Bowman's capsule or renal corpuscle, a part of the renal corpuscle. Second step is tubular reabsorption. That is second step, tubular reabsorption from fluid into the blood. Third step is tubular secretion. Additional wastes, drug and excess ions are actively secreted into the renal tubule. Among these three processes, three steps, glomerular filtration is the very first and most critical step, because without filtration no urine can be formed. So this is the details of filtration membrane. The number one is the fenestration or pore of glomerular endothelial cell, prevents filtration of blood cells but allows all components of blood plasma to pass through. Second one is the basal lamina of glomerulus, prevents filtration of larger proteins. Third is the slit membrane between pedicels, prevent filtration of medium sized proteins. This is the filtration slit, or this is the pedicel, podocyte of the visceral layer of glomerular or Bowman's capsule. What is GFR? Why is GFR clinically important? Doctors routinely estimate GFR because it is the best indicator of kidney function. This is the transmission electron microscopy image of the filtration membrane showing the pedicel of podocyte, filtration slit, lumen of glomerulus, and this is the fenestration or pore of glomerular endothelial cells. So when high GFR may occur during pregnancy, high protein intake, or early diabetes. Low GFR occurs in chronic kidney disease, acute kidney injury, severe dehydration, renal artery obstruction. Therefore, monitoring GFR helps detect kidney disease even before symptoms appear. So we have to know factors affecting GFR. Now let's discuss the major factors regulating GFR. Number one is renal blood flow. More blood reaches the kidneys, more filtration occurs. Therefore, GFR is directly proportional to renal blood flow. Normal renal blood flow is about 1.3 liters per minute. Tubuloglomerular feedback. This is an autoregulatory mechanism. The macula densa detects sodium chloride concentration in the distal tubule. If sodium chloride increases, the macula densa signals constriction of the afferent arteriole, reducing GFR. If sodium chloride decreases, the afferent arteriole dilates, increasing GFR. This mechanism keeps the filtration remarkably stable. Third point is the glomerular capillary pressure. Normal pressure is approximately 60 mmHg. Higher pressure produces greater filtration. Therefore, GFR increases with glomerular capillary pressure. Fourth is the plasma colloid osmotic pressure. This pressure is generated by plasma proteins. Normal value is 25 mmHg, and increase in plasma proteins pull water back into the capillaries, thus reducing GFR. Fifth point is hydrostatic pressure in Bowman's capsule. Normal value is 15 mmHg. This pressure opposes filtration. Conditions like urinary tract obstruction increases Bowman's capsule pressure, thereby decreasing GFR. Afferent arteriolar constriction. Constriction reduces blood entering the glomerulus. Less blood means less filtration, so GFR decreases. Efferent arteriolar constriction. Initially, blood accumulates inside the glomerulus, raising the filtration process. Therefore, GFR initially increases. However, prolonged constriction eventually decreases GFR because fresh blood cannot enter efficiently. Systemic blood pressure. The kidneys possesses autoregulation. As long as arterial pressure remains between 60 and 180 mmHg, GFR remains almost constant. Outside this range, GFR changes significantly. Sympathetic stimulation. Mild stimulation produces little effect. However, strong sympathetic activity such as during severe stress or hemorrhage causes vasoconstriction, reducing renal blood flow and GFR. Surface area of filtration membrane. Diseases damaging the glomerulus reduce filtration surface area, leading to reduced GFR. Permeability of filtration membrane. In inflammation or hypoxia, membrane permeability increases. As a result, even plasma proteins appear in urine, a condition known as proteinuria. Mesangial cell contraction. Contraction of mesangial cells decreases filtration surface area, thus lowering GFR. Hormonal regulation. Hormones increasing GFR. Atrial natriuretic peptide or ANP, brain natriuretic peptide or BNP, dopamine, nitric oxide, PGE2. These promote vasodilation. Hormones that decreasing GFR: angiotensin 2, endothelin, noradrenaline, platelet activating factor, platelet derived growth factor or PGF2 alpha. This may cause vasoconstriction. So this is the image showing the pressures that drive glomerular filtration. Number one is the glomerular blood hydrostatic pressure. GBHP is approximately 55 mmHg. Second one is the glomerular hydrostatic pressure or CHP, that is 15 mmHg. And third component is the blood colloid osmotic pressure, that is BCOP, or 30 mmHg. So the net filtration pressure or NFP from the glomerular filtration bed will be GBHP minus CHP minus BCOP, or 55 mmHg minus 15 mmHg minus 30 mmHg is equal to 10 mmHg. So summary, let's revise. Urine formation has three basic steps: glomerular filtration, tubular reabsorption and tubular secretion. Normal GFR is 125 ml per minute or 180 liters per day. Only about 1 to 1.5 liters are excreted as urine. So GFR mainly depends on renal blood flow, glomerular pressure, plasma oncotic pressure, Bowman's capsule pressure, afferent and efferent arteriolar diameter, tubuloglomerular feedback and hormonal regulation. So, glomerular filtration is the foundation of urine formation and one of the most precisely regulated processes in human physiology. A healthy GFR ensures that the kidneys effectively remove waste products while preserving essential nutrients and maintaining the body's internal environment. Understanding GFR is essentially not only for examinations like B.Sc, M.Sc, NEET, nursing and medical biology, but also for understanding how kidney disease develop and how they are diagnosed in clinical practice. If you found this class helpful, please like, share and subscribe to Biology at Priyajit for more easy to understand biology and human physiology classes. Thank you for watching. Keep learning, keep growing, and I will see you in the next class.